Sorry. Um, okay. You can mark on this whiteboard too, right? On my whiteboard, that can you see my whiteboard?
Yeah.
You can write on this too, right?
No.
Mm. Didn't I get it one time where you were able to do it too?
Yeah. It was like on the side. On my screen, it would show it like on the side, but there's nothing here.
Mm. And you don't have anything on yours, right?
No.
Are you sure it's not something with your computer? Because how are you able to do it last time, but you can't do it this time? Oh,
Mm.
no. Mhm. Mm. Hey Liz, can you draw on this whiteboard or no?
Can I draw?
Yeah.
Let me see. Yeah, I can. Did you see that?
Yeah, I can see it.
Okay.
Well, it's not letting me.
Why?
I don't know.
Is that you, Bridgie?
Where?
The
No,
green?
no, this is me.
No.
Oh,
Like it would usually show me like a pen or something, and I don't see anything.
like on the bottom, Bridgie? Or on the top?
It would be on like on the side, on the left side for me or something.
That's not right.
Is it my computer? Like, am I supposed to enable something or something?
We didn't do it last time?
I don't remember what we had to do last time. Last time I remember just doing the whiteboard and then it worked.
Yeah.
Because right now she can't share. I want her to be able to share her screen too.
Yeah.
Multiple. Who can start sharing when someone else is sharing?
Dr. Magic, Dr. Beach said that you're a very kind man.
Well, thank you. Open and collaborate. It says everybody can edit. How about now?
Okay, now I can.
Okay.
Can you
So it
see
was
it?
you all this time, Dr. Magic. You had us blocked,
I didn't have anybody blocked, okay?
like
I'm
on
trying to,
Facebook. Like Dr. Ansari has us blocked.
nobody has anybody blocked. See here. I want to see if I can hear. Can I control view? thought I could. I thought I could paste the thing. Hybrid, try sharing your screen now. See what happens. Like I'm gonna share my screen. I'll show you what I see. You can see my entire screen, right?
Yeah.
Can you see? Actually, no, you can't see that. Anyways, dude, my mouse is being really annoying. Okay. Yeah, I wanted you to kind of show me and walk me through this whole pedigree analysis like I just did, but does it make sense what we did?
Yeah.
You think you could do it on your own?
Yeah. I could like you said, I could like explain it to my mom when she gets here or something.
Yeah, do that. I wanted you to explain to me, but I don't know why I can't get this to work. So, but really, that was the last big thing here. Does this chart make sense? I mean, this is just some BS that you put in. That's not important. Does this right here is important? Does this make sense?
Mm.
There's all different types of genetic patterns that you can have. And we
What
talked
is
about,
what is called dominance and pleiotropy, something like that. Pleo something.
so let's go through this one at a time. Complete dominance of one allele. That's normal, right?
Yeah.
If there's a big P, that's going to show. So purple shows when there's just one big P or two big P's, right? Makes sense?
Yeah.
Incomplete dominance means um,
Is that when it doesn't, when it shows like mixed?
right.
Like, if red is dominant and white is recessive,
And then there's
but then
pink.
the flower's like pink.
Well, white isn't recessive. They're both dominant. They're not. There's incomplete dominance. So it's like you have C, C. One is R superscript
Hmm.
and one is W superscript. So just like they have it here. This mouse is really annoying me right now. So you have C. Um, R and C W. Okay. So when you have just R C R C R, it's red. When you have just C W C W, it's white. And when you have C
Hmm.
R C W, you get pink in between.
Okay.
Okay. You get a phenotype that wasn't present previously. Codominance means that both phenotypes show up. Okay. So you have an A and a B, and when they're both together, you get AB blood. Okay. So
Okay.
how is that different from incomplete dominance? Incomplete dominance, you get a mixture. You have red, white, and pink. If it was codominance, you'd have parts of the flower that were red, like a streak of red, a streak of white, a streak of red, a streak of white, but you don't see that. You see
Oh,
red,
okay.
white, and then pink. They mix.
So it blends and the incomplete.
Yeah, it blends. You get a phenotype that wasn't previously there. In codominance, they both show completely. You'll get on the same flower, you'll get a section that's red and a section that's white
Yeah.
because they're both dominant. They both show. And in complete dominance, they don't. They're both not completely dominant over the other. That's why they kind of just blend.
Okay.
Multiple alleles. We saw that with blood types. You have A, B, and O, right?
Yeah.
Um, Pleiotropy. one gene affects multiple phenotypic characteristics. Um, I want to see an example of that here. Where should talk about that here? Okay, pleiotropy is where, you know how typically we had one gene affecting one characteristic?
Mm-hmm.
So big A means you have, or big P means you have purple flowers.
Yeah.
So pleiotropy just means that big P not only means you have purple flowers, but it means you have um, circular flowers instead of like triangle shaped flowers. So
Okay.
one gene has multiple phenotypic characteristics.
Oh, okay.
So one gene has multiple phenotypes, affects multiple phenotypic characters. So one gene can affect not only the color, but it can also affect shape, essentially.
Okay.
Epistasis means that one gene is kind of like the master switch, and if it's on, it causes the rest to be on. And if it's off, even if the other ones are on, they won't turn on. Kind of like in this hotel room, we have a master light switch. I can turn all the lamps here on the on position, but if that master light switch is off, none of these are going to turn on.
Okay.
When I have the master light switch on, then these lights will be on only if I turn these lights on. So even if the lights are on individually, the master light switch still needs to be on. But if the master light switch is off, I can keep all these lamps on. None of them will turn on because the master switch is off.
Okay.
That's what epistasis is. And then polygenic is the opposite of pleiotropy. So we said one gene can affect multiple things.
Mm-hmm.
Big P can mean purple and round flowers. Here, in order to get, I don't know, tall flowers, you need two genes. So
Yeah.
multiple genes are involved to affect some type of phenotype. In order to get really tall flowers, I need two genes to be active, or I need two genes to be working together to give me something really tall. So we had one gene affecting multiple phenotypes. Here we have multiple genes contributing to one phenotype.
Okay.
So A, B, and C will all affect color. So see how we have different shades? It starts off as like blonde up here and like really dark brown in the bottom corner.
Yeah.
Shades in between
Mm-hmm.
contribute to affect one phenotype. It's polygenic. So the trait for like skin color is polygenic. We have multiple genes that contribute to affect skin color. Does that make sense?
I couldn't hear your audio went like off.
Yeah.
I was just saying, polygenic means we have multiple genes affecting one phenotype. So here, it's like skin color. See how in the top left corner we have white, and in the bottom right we have brown, like really dark brown.
Yeah.
So we need multiple genes contribute to skin color. So
Mm-hmm.
how the genes interact with each other, we'll get different shades of color, and that's how we have different like variations in skin tone, for instance.
Okay.
So in pleiotropy, we had one gene affecting multiple phenotypes. Big P means purple flowers, and it means also it's affecting the shape. It's
Mhm.
affecting color and shape. Here we have multiple genes all contributing to just one phenotype, which is the color.
Oh, okay.
We have a whole spectrum of color. We can be like white or we can be dark brown. How do we, and then we have all sorts of shades in between. It all just depends on if we have big A, big B, little b, like what combination of that mixes together to give us skin color.
Okay.
So it's polygenic. Multiple genes contribute to give us like a final color, a final skin.
Okay.
It's not just one gene controlled. It's like when you get multiple genes from it.
Okay.
See polygenic right here?
Mhm.
Two or more genes act additively to produce a phenotype. Human height, skin color. So you need the effect of multiple genes. Pleiotropy. One gene affects multiple aspects of a phenotype. So multifactorial diseases, it could be the result of multiple genes plus environmental factors. Like they're exposed to radiation or they're exposed to something, and it had to do with their genes too. So it's multifactorial. Lots of factors.
Okay.
I think
Okay.
I read that there was gonna be a bunch of Hardy Weinberg problems.
Okay. Do you have okay. I think did she ever give you the answers to these questions right here, Bridget? That are on this
Um,
slide?
I don't remember because sometimes she puts it on the in a video.
Because I can give you what I think are the answers, but the reason I ask is just because I wanted a way to double check it.
Yeah, let me check.
So this is pretty much what you need to know right here.
Frequency of the allele.
Yeah. Because it's saying for a gene for hair color, you're either gonna have
Mm.
it's either gonna be big R or little r.
Mhm.
Big R means it's red, little r means it's black.
Mhm.
It's always going to be one of these two. The one allele is going to be this. The allele is either going to be this or it's going to be this. Okay?
Mhm.
So 100% of the time, 100% of the time, it's either going to be big R
Or
or it's
little
going to.
r.
So if big R shows up 75% of the time, little r has to show up 25% of the time.
Is it always like that?
Not always. I'm just saying they always have to equal 100,
Yeah.
right? Or the other way to say it is 1. Their
Mhm.
frequency is always 1. Because instead of saying 75%, if we put it in decimals, it'd be
Oh yeah, 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 yeah.
0.75. Because there's only two alleles.
Yeah.
So if one shows up 0.75% of the time, the other one shows up 0.25. Now, they're making certain assumptions, which, like she said, is never true of a population, but we just make certain assumptions. Just like a lot of times when we're doing physics problems, we say, well, let's ignore friction, let's ignore um, wind resistance and stuff like that. So these are the 7 things that you have to assume when you're doing Heidi Weinberg.
Okay.
Okay. And it's never fully true for any population, but based off of that, you can make some certain assumptions. Okay.
Okay.
The dominant allele is P and the recessive allele is Q. So here, if you know,
Dogs heard me. That's why.
dogs heard you. So
I'm
we know,
parking, so they heard the car.
so we know P plus Q is equal to 1. Right? That's what I just said. 100% of the time, it's either going to be big R or little r, right? 100% of the time. Because we only have 2 alleles for this gene, either big R or little r. There's no other allele, right?
Yeah.
So 100% of the time, it's either going to be big R or little r. And when we put percentages in decimals, we just we get like point whatever, right? Because we just move everything over 2 decimal spots. So it's 1. Because like 25% in decimals is how much? 0.25, right? You with me, Richard?
Yeah.
Does that make sense?
Mm-hmm.
So you just make P the dominant allele and Q the small allele, okay? Now,
Okay.
when we do the Punnett square, right? Because um, in a heterozygous person, like let's say it's big R,
Yeah.
little r, right? Big R, little r, okay? Big R times big R and big R, we'd have that here, right? Big R, right?
Yeah.
If we said R was the P was equal to our dominant allele, that's P times P, which is P squared, right?
Mm-hmm.
P2 is not P2, it's P squared.
Okay.
And then PQ and PQ. PQ plus PQ is 2PQ, right?
Uh-huh.
That's what that is right here.
Oh.
And then this is like our little r from our Punnett square. If we said little r was the Q, Q was a small recessive allele, that's like little r, that's Q times Q, which is Q squared. So P squared plus 2PQ plus Q squared is equal to 1. Q squared is our dominant homozygous. Q squared is our recessive homozygous. And then 2PQ is our heterozygous. It shows up 50% of the time right here.
Oh, okay.
Does that make sense?
Yeah.
So now they're saying allele B is present in a large random mating population at a frequency of 54%. Okay?
Okay.
So let's see what we have to do here. We have to do some math.
Were you able to find the answers?
Okay.
Did
So
you
here's the
post-
thing. We have two equations.
Yeah, I was
Okay.
gonna.
Do you know the rule about variables?
What's the rule?
The rule is if you want to solve, if you have two variables and you want to figure out what they are, you have to have two equations that relate the two variables together. So we have P plus Q is equal to 1, right?
Mhm.
P is how often the dominant allele shows up. Q is how often the recessive allele shows up. And then we also have P squared plus 2PQ plus what?
Q2.
Q squared, right?
Yeah, square.
Is equal to 1, right?
Mm-hmm.
So right now we don't have a way to solve for this. So whenever you have two variables, you need two equations that relate the two variables together. When I say relate the two variables together, meaning two equations where the two variables are in both equations. Because what we do is we solve for one variable in terms of the other and then plug it into the other equation. So you get P is equal to 1 minus Q, right? I just moved this over to this side. See how I did that?
Mm-hmm.
P is equal to 1 minus Q. Then I just take this whole term and I substitute it in over here. So wherever I have P, I now just have 1 minus Q
Oh, squared.
squared plus 2 times P, which we said P is equal to 1 minus Q times Q plus Q squared is equal to 1 plus Q squared is equal to 1. Now this whole equation is in terms of Q. Now we just solve for Q. But anyways, that's some excessive stuff that we don't need. What we need is the basic two equations that they gave us. What are those basic two equations? This right here,
It's Q.
Mm-hmm.
P plus Q is 1, and then P squared plus 2PQ plus Q squared is equal to 1. So what I want you to pay attention to is this equation is for genotype frequency, okay?
Mm-hmm.
So how often will we get AA? Well,
Well, often.
AA is going to be equal to P squared, okay?
Mm-hmm.
Does that make sense
Oh,
why?
so once.
And then for allele frequency, if I just want to figure out how often the alleles show up, that's going to be equal to P plus Q equals 1. Okay? Does that make sense why?
I got confused.
You had me at the p squared and the 2a's.
So I said for a gene that has two alleles, we're gonna have a dominant version, A,
Mhm.
and a recessive version, little a, right?
Mhm.
Now the first thing that we know is that there's only those two alleles, right? Every single time I look for at that gene, I'm only gonna have one of the two alleles. Either big A, the dominant version, or little a, the recessive version, right?
Mhm.
100% of the time, every single time I look, I'm gonna find one of the two, right?
Right.
Mhm.
Either big A or little a, okay? So that should make sense. Every time I look at this gene, I'm either gonna find big A or little a. If I have more big A, then I'm gonna find little a less, right? Because
Yeah.
every single time I look, I'm either gonna find big A or little a. So big
Mhm.
A shows up 75% of the time, 0.75. The other 25% of the time, it has to be little a. Or the opposite is true. If little a is present, 50% of the time, big A has to be present the rest of the time, right?
Yeah.
Because I can only find one of the two. I'm not gonna find like, I don't know, medium A or something weird like extra large A. I'm not gonna find that. It's just gonna be big A or little A. If I see big A 50% of the time, I'm gonna see little a 50% of the time. If I see big A 75% of the time, I'm gonna see little a 25% of the time. Because
Right.
together they have to equal one.
Mhm.
Like every single time, I'm gonna see only one of the two. If I see more of one, I'm gonna see less of the other. If I see
Yeah.
more of the other, I'm gonna see less of the first one. Does that make sense?
Yeah.
So that's what this first equation is telling you, allele frequency. Okay? It just randomly, it just randomly called P the dominant allele. It made that the variable for the dominant allele. Okay?
Okay.
And it randomly assigned Q as a variable for the recessive allele. Okay?
Okay.
So now this is talking about genotype frequencies for for a Punnett square of two heterozygotes. So pretend we had big A, little a, big A, little a.
Where did that?
Okay?
Mhm.
And we do the Punnett square. What do we get over here? Big A, right?
Yeah.
Now, over here, we just said P is equal to what?
The big A?
Good.
The-
P is equal to big A. So if big A is here and P is equal to big A, it's P times P or P squared,
square.
Mhm.
Okay.
right? Over here in this small box, this box down here, I have little a, right?
Yeah.
And we said Q is equal to the recessive allele. So it's
Yeah.
like Q times Q, which is Q squared.
Mhm.
Then over here, I have big A, little a, big A,
Okay.
little a.
And then little A. Okay, I
So
got
this is like P times Q, right?
Mm-hmm.
Mhm.
There's P times Q. And in this box right here, I have P times Q, right?
Yeah.
Mhm.
So it's just telling me that I'm gonna and then this is gonna be P times Q plus P times Q is 2PQ, right?
Yeah.
So
PQ.
when I cross a heterozygote for this genotype right here, big A little a times
Mm-hmm.
big A little a, I'm only gonna get one of three options. Either this one, or this one, right?
Yeah.
Mhm.
So this is just telling me what my probabilities are of getting this one. What percentage am I going to get this one? It's P squared. What percentage am I going to get? This one? It's 2PQ. What
Oh.
percentage am I going to get this one? It's Q squared.
Mhm.
Does that make sense?
Yeah.
Yeah, that makes sense.
Okay, so now let's run through this problem. So it says allele B, okay? Allele B. Is this capital or
Yes.
lowercase?
Capital.
Capital, right? So it's the big one, it's the dominant one. Now again, I don't want you to think that because it's dominant, it's gonna be more frequent. That's not the case, okay? Something can be dominant but be rare, okay? Make sure you understand that. Just because it's dominant doesn't mean it shows up all the time. When it's
Yeah.
dominant, it means when it shows up, it wins the battle. But it doesn't mean that it's always present, okay?
Okay.
Just like a skunk. A skunk, when it's present, its smell makes everything smelly, but it's not always around, right?
Yeah.
The
Okay.
smell is very dominant, but it doesn't mean it's very frequent,
Okay.
okay? A skunk isn't around all the time.
Yeah.
But when it is, we smell it very strongly, right?
Yeah.
So it's think of it, it's dominant, but it doesn't mean that it's always around. It doesn't mean it's always present. So just because it's dominant doesn't mean that it's always going to be like 70%, or it can be found 3% of the time. The dominant allele can be found only 3% of the time. Very, very little bit. But in those 3%, when it's found, it's going to have a very strong effect. Dominant
Okay.
just means it has a strong effect. It doesn't mean it's always around. Okay?
Okay.
So now, allele B, which is the dominant allele, is present in a large random mating population. That's just to tell you all the Hardy-Weinberg assumptions from above, those 7 things, that they're all present. So we can solve this problem using the Hardy-Weinberg equations, which is this guy and this guy. Okay?
Okay.
At a frequency of 54%. So it's saying the allele frequency. Allele frequency. So which equation do we have to use for allele frequency?
The P plus Q equals
P
1.
plus Q. Now, the dominant allele, is that P or is that Q?
What did you say?
The dominant allele, is that P or is that Q?
P.
P.
P. Okay. So 54%. So we just have to convert that into decimals, right? How do you convert that to decimals?
Divide by 100.
Yeah. Or you literally just, a
Move
trick is
forward.
just to move that decimal place over 2 times.
0.54.
So 0.54, right?
Yeah.
Plus Q is equal to 1. So what does Q equal?
Wait,
1
what?
minus 0.54, so Q is equal to 0.4